Good morning, class. Previously, we finished a lecture on the physiology of the male and the female reproductive system. We will now want to go into a lecture on the development of the embryo and the placenta. We all know that the embryo is a fusion between the male and the female gamete, and this leads to the formation of a zygote. The zygote has to implant in the endometrium. That is, its specific implantation site is on or in either the anterior or the posterior wall of the uterus, specifically in the endometrium. After that, what happens in the ensuing days? What nourishes, or how is the conceptus nourished? How does the baby develop, or the fetus develop? And the end results, we are going to see how we can deliver a viable fetus or a viable baby through the placenta. So stay tuned as we begin the lecture. Thank you. Development of the embryo and the placenta. So the lecture will cover, or it will explain the main events that occur during the first 4 weeks of development. It will also evaluate how the placenta evolves, its formation and its functions during the first month for the development and maintenance of pregnancy under normal conditions. And this requires the attention and the competence of general practitioners. So the placenta starts development in the 4th month of pregnancy. 4th month of pregnancy. We also interpret the functions and alterations of the placenta. Okay, so let's begin. Fertilization is the process by which male and female gametes fuse or combine, and it occurs in the ampullary region of the uterine tube. And the ampullary region is where most of the ectopic pregnancies occur. And I showed you the ampullary region when we were doing the anatomy of the female reproductive system, the womb, the uterus. Okay, so the sperm cannot fertilize the oocyte just after reaching the female reproductive tract, but must undergo 1, capacitation, and 2, a chromosome reaction to acquire this ability. This is followed by after fertilization and zygote formation, it is followed by cleavage or segmentation. Then the cleavage or segmentation will lead to the formation of a blastocyst, and which in the process called blastulation. Then the blastocyst will go and implant in the endometrium. It will go and implant in the endometrium, the functional layer of the endometrium. So let's watch a short video on how this fertilization process happens up to segmentation and blastocyst formation. Blastocyst. That's the conceptus that implants, or the embryo that implants in, but at this stage it's not an embryo, it's a blastocyst. This is what implants in the endometrium, in the functional layer of the endometrium. So this implants or enters into the endometrium, implants in the functional layer of the endometrium. Layer of the endometrium. Endometrium. Okay, so that's the endometrium, like I explained in the lecture on anatomy of the female reproductive system. This is where the fetus, this is the segment of the uterus or the womb where the conceptus or blastocyst implants. So let's watch a video on this event. Let's watch a video. Let's watch a video on this event. So let's watch a video on this event. But before we watch the video, before we watch this video, we would like to give acknowledgement to the original owners of the various videos that we are about to show on this lecture, because it took them a whole lot of strength, agility, frantic effort to follow through with these videos. So we'd like to acknowledge them because they have the right, or they have the copyright. They are the copyright holders of this video. So we want to acknowledge them for this wonderful work that they have done. Let's zero in on a video on fertilization, then blastulation, blastocyst formation.
When ejaculation occurs during intercourse, approximately 200 million sperm, or spermatozoa, are deposited into the vagina. They swim through the cervix, propelled by whip-like motions of their tails, or flagella, after which muscular contractions of the uterus direct them to the uterine tubes. This process usually takes between 30 minutes and 2 hours. Only around 200 spermatozoa will reach the secondary oocyte in the uterine tube, and of these, only one will fertilize it. Fertilization cannot occur until two processes have taken place: capacitation and the acrosomal reaction. These can take several hours. Capacitation is not fully understood, but secretions from the uterus wall and uterine tube destabilize the plasma membrane surrounding the head of the spermatozoa, or acrosome, resulting in the membrane becoming more fluid, which helps to prepare the spermatozoa for the events of fertilization. The spermatozoa become hyperactive. Their flagella beat more frequently and their heads move laterally. The capacitated spermatozoa move through the corona radiata, a dense layer of granulosa cells surrounding the oocyte, and come into contact with the zona pellucida. The zona pellucida expresses specific receptor proteins called ZP3, which bind to proteins expressed in the heads of the spermatozoa. The binding of ZP3 triggers the acrosome reaction, during which the enzymatic contents of the acrosome are released. These enzymes help to digest a path through the zona pellucida, allowing the spermatozoa to enter the perivitelline space and reach the plasma membrane of the secondary oocyte with which it fuses. To ensure that only one spermatozoan penetrates the zona pellucida and fuses with the oocyte membrane, fusion of the spermatozoan and oocyte membranes activates a fast and a slow block to polyspermy. During fast block to polyspermy, after fusion, the oocyte membrane depolarizes, preventing other spermatozoa from fusing with it. Slow block to polyspermy is also stimulated by this depolarization. During slow block to polyspermy, a wave of intracellular calcium is released, causing small cortical granules beneath the oocyte membrane to release their contents, rendering ZP3 inactive and making the zona pellucida impermeable. Upon the spermatozoan entering, the oocyte undergoes meiosis II and further develops into the female pronucleus. During this time, the sperm develops into the male pronucleus, and the two pronuclei fuse to form a single diploid nucleus, or zygote.
Okay, so we are back. So let's see. So fertilization is the process by which the male and female gametes unite and give rise to the egg or zygote. No, it is the process by which the male and female gametes unite and give rise to the zygote, not the egg. Begin the formation of a new individual. Okay, so zygote is produced, not egg. So if you see it in your slide, correct it. It is a crucial stage of development in which there is an interaction between both gametes, with the consequent fusion and mixture of paternal and maternal hereditary characteristics. So, let's see. So, we have, if you have a female, okay, a male first. This is a male. Male. It is macho hands. A male has 23 chromosomes. That is the germ cells. Germ cells. Or sex chromosomes. A female with long earrings and some Rosalinda flowers on her hair. Beautiful female. Okay, so a female also has 23 sex chromosomes. This one does not have matches. This is Bolle. So sex chromosomes, female also has so 23. So will lead to so this is a haploid or half. When we say, if every normal human being has a diploid number of chromosomes, diploid, which is what? 46 into bracket 2n or 46. 46 chromosomes. But the haploid is 23, n or 23 chromosomes. This is n or 23 chromosomes. So this is half of the 46, because every normal human being has 26 diploid chromosomes. But the sex ones that you produce, we have what? 46. We have what? The man will produce what? 23 sets. The woman will bring 23. The man will bring 23. To become what? So 23 plus 23 will lead to a new individual. Oh, sorry. This is a new human being. A new human being. Baby boy having what? 46 chromosomes. 46 or 2n. 2n means haploid. 2 times the haploid. So it means diploid. So diploid. Diploid means double. So double of the 23, giving us diploid chromosomes. Diploid. Okay, so this is the human being we have. We give birth to this the baby with what? Half of the genes of the mother and half from the father. So father half, mother half, giving what? A baby with 46 chromosomes. Let's continue. So where does fertilization occur? In the human species, fertilization is internal. It occurs in the uterus. It is found in what? The external third of the uterus, or the amplar region. So the amplar region of the uterus. So ampullary region of fallopian tube. So let's see. So this is the amplar region of fallopian tube. This is the fallopian tube. Let's see more images of the amplar region of the fallopian tube. So if this is the fallopian tube, this is the amplar region. We have the first, this finger-like projections is called fimbriae, infundibulum, ampulla. Then this side is the isthmus. So let's get a complete image which is big, a bigger image. Okay, so let's see. It seems this one is clearer. Let's see. So we have an image like this. So this is the, no, this is not what I want. Okay, so this is the fimbriae, the ovary, the fimbriae, infundibulum. This section from here to here is the amplar region. Amplar region. Another one is here. So that's the amplar region. This is the main image. This is the amplar region. Okay, this is clearer. Fertilization occurs in the amplar region, this side. This is the fundibulum, the finger-like projection. This fertilization. This egg is now fertilized. So this is all this side from here to here is the ampulla, and this is the isthmus. So let's go back to, so this is the ampullary region. This is the isthmus. Isthmus is closer to the uterus. So this is the fundibulum and the fimbriae. Okay, so let's go back. So where does fertilization occur in the ampullar region? Sperm cannot fertilize the oocyte just after reaching the female reproductive tract, but must undergo capacitation and acrosome reaction like were explained in the video. So let me throw a little light on it. So capacitation is the acquisition by the sperm of the ability to fertilize. Thus, the sperm is capacitated, given the capacity to be able to fertilize. And this process occurs in the female reproductive tract when the covering of the glycoproteins and plasma proteins on the cell surface are removed, and this allowing the entrance of the spermatozoa into the corona radiata. Capacitation promotes sperm motility and is a necessary preparation for the acrosome reaction. So let's go. Let me explain capacitation. So this is what these cells around the ovum. This is the ovum or the egg. The cells around it are called corona radiata. Corona radiata. Then this boundary, this white boundary, is what is called zona pellucida. Zona pellucida. Then this is a polar body. I will explain that one later. We have the secondary oocyte. Then the secondary, this is the egg. The secondary oocyte is the egg. The primary oocyte develops into the secondary oocyte. Then we have the meiotic spindle. So this is the meiotic spindle. So this is what happens when an egg, a sperm penetrates the egg. And when this sperm is, one sperm is able to penetrate the plasma membrane. This is the plasma membrane. When one sperm is able to penetrate this plasma membrane, only one sperm will penetrate this plasma membrane. Then the egg will say that no, I will not allow any other sperm to come and fertilize me again. So the boundary will be closed. The zona pellucida will now be resistant, so no other sperm can enter. That is nature. Because if another sperm enter and fertilize again, and another sperm, this will end up in Siamese strings and a whole lot of congenital anomalies. So only one sperm should fertilize an egg. No greediness. So these are corona radiata, follicle. Corona radiators are follicular cells layer surrounding the secondary oocyte. We call this one secondary oocyte. You know, the egg comes as an oogonia. Initially we call it oogonia. Oogonia, as you saw in your physiology of the female reproductive system. So the egg is an oogonia. Oogonia. That is in its crude form or immature form. Then the sperm also, if it's not matured, it is called what do we call it? We have the oogonia for the egg, and also so like I said, the oogonia is the primitive form of, or the immature form of the female ovum or egg, as the spermatogonia or spermatogonium, if it's singular, is the immature form of the male gamete, or yes, the male gamete, or the sperm. Okay, that's spermatogonium, oogonia. Okay, so this one produces spermatocytes, and this one produces oocytes. Oocytes. As a mature form, this one produces spermatocyte. That makes spermatocyte secondary spermatocyte. So immature form leading to mature form, immature form of the egg, begonia leading to mature form oocyte. Zona pellucida is a glycoprotein layer surrounding ovum. So this is the zona pellucida. It's composed of glycoprotein. This white boundary is composed of glycoprotein. I know I taught you glycoprotein. Okay, so overview of capacitation. So how is the sperm capacitated or strengthened or activated to be able to move freely and also energetically in a rapid linear progression? Let's see. So there's a cholesterol coat around the sperm cell, and this the uterus or the womb, the fallopian tubes and the rest, the cervix and the rest, especially the fallopian tubes, the fluid and those things, they react with the cholesterol coat and removes the cholesterol coat, so that the sperm can be free enough to swim through the vagina, through the cervix, through the uterus, into the amplar region of the fallopian tube, where there is what? Fertilization. Okay, so let's also look at the acrosome reaction. So acrosome reaction, you see this is the acrosome reaction. So the sperm binds onto the zona pellucida. The yellow boundary is the zona pellucida. It's jelly, it has a jelly coat. The sperm bounds onto it. The coronal radiata cells are around it. Then this is the inside. This reddish color or orange color is the egg cell or ovum. Egg cell is also called ovum. So when the sperm touches, the head of the sperm touches, and the head of the sperm contains something we call acrosome. Acrosome, we'll see it later. Acrosome. So when it touches the zona pellucida, this yellow thing, this is the outside. This is the zona pellucida, the yellowish, this yellowish thing is the zona pellucida, and this is the egg. Inside this, the orange color. So when the egg, and this is the sperm, and this is the acrosome. This yellow thing here on the head of the sperm is the acrosome. And this is the genetic material or cytoplasm of the sperm. So when the sperm touches, it uses its acrosome layer to touch the zona pellucida. What happens? There is binding of the sperm to the jelly-like coat, that's zona pellucida. So enzymes are released from the head of the sperm or the acrosome to react with the zona pellucida. Enzymes digest the zona pellucida. You can see the yellowish thing has been digested. Then, after digesting it, it releases its genetic material. So the nucleus enters the egg cell, and this process is what we call the acrosome reaction. The acrosome reaction is the reaction of the acrosome layer with the zona pellucida to end up in the releasing of the nucleus into the cytoplasm of the sperm cell. Okay, so let's also see the cortical reaction. Cortical reaction is the reaction that occurs in order to prevent polyspermy. That means that when one egg enters the zona pellucida and fertilizes the egg, then the other sperms are excluded or prevented from entering. And this will prevent what we call polyspermy. It will prevent polyspermy. To prevent that's multiple sperms fertilizing one ovum or one egg. It's a taboo. So we want to prevent polyspermy. So this is what we do. Yes, so the egg will enter. This is the cortical granule. It's found inside the ovum or the egg cell. Then there is a sperm binding protein. There is a protein inside the ovum where the sperm binds. So that's the zona pellucida. So the sperm will use its head, the acrosome there, to bind to the zona pellucida. And the zona pellucida has a sperm binding protein. It's like a peg and a hole, or a key and a lock and key model. So after binding, no other sperm can be able to bind, because this acrosome layer, you can see the zona pellucida has been hardened, so no sperm can enter. But here it is soft. That's why you are seeing it zigzag, rough character or characteristics. Now it is very firm, and this will prevent multiple sperms from fertilizing the same egg. So the phases of fertilization is penetration of the corona radiata, that's the cells that surround the egg or the zona pellucida. Union and penetration of the zona pellucida, yes, the boundary is penetrated by the sperm, one sperm. And when one sperm penetrates it, no other sperm can penetrate again to fertilize the egg. Union and fusion of the sperm to the cell membrane of the egg. When the sperm enters, or the nucleus enters the nucleus of the egg, women are always waiting to receive something from man. It also occurs in the molecular level or the cellular level. The sperm has to swim. Women, after swimming into the ampler region of the uterine tube or the fallopian tube, it now has to go and enter the zona pellucida, enter the perivitelline space, enter the plasma membrane of the egg, then enter the cytoplasm. Then it will go and now fuse with the pronucleus of the female. What a long journey. Men are always suffering. So after entering, there is prevention of additional sperms from entering. So that is what we call polyspermy. Metabolic activation of the egg. The egg now has a lot of mitochondria and the rest. So it will now cook food. The sperm travel to the egg. The sperm is so tired, so the egg now has to prepare food, so release energy to take care of the union. So if you go and bring a woman to your house, you give her money, she will now prepare food for you, so there will be food for the two of you during the honeymoon. Decondensation of the sperm nucleus. So the sperm now becomes a male pronucleus, or the genetic material becomes activated. There is completion of the meiosis. Now the sperm cell now, the egg cell now completes its meiosis, because in the uterus, the egg, the fetus, or the female sex fetus in the uterus, or female baby in the uterus, what it does is that it starts, let's say, it starts producing gametes. So it starts its meiosis, mitosis, meiosis, producing gametes. Then it ends in meiosis 2, metaphase of meiosis 2. Then it will stop. So it produces the egg. So the fetus in the womb start producing egg. Even by 3 months' time in the womb, it starts producing eggs. But the eggs are not activated. They are only activated during puberty, when because it will produce the egg. The eggs will not be active. The eggs will undergo division of their genetic material, whatever. Then it will now stop at meiosis 2, metaphase of meiosis 2. When it stops there until puberty, then it will start releasing the egg. in menstruation. So, or when it meets a sperm, the egg will now complete its meiotic division in meiosis 2, metaphase of meiosis 2. So God is so wonderful. Women, when they are even in the womb, they produce eggs, but the eggs are not matured. They mature only at puberty, when they can now give birth. Development of the female pronucleus. Contact of the pronuclei and the mixture of paternal and maternal chromosomes. So when the two pronucleus, or the two nucleus, the female and the male nucleus, join together, they form a zygote. Okay. Organization and preparation for the first mitotic division. So after forming the zygote will now undergo mitosis. It will divide several times to form the 8-cell stage, the 16-cell stage, which is morula, 32-cell stage, which is the blastocyst, and implant in the meiotrium. Cytoplasmic segregation before cleavage. So the cytoplasm will be separated into pieces before the zygote can divide. So let's see the events. So phase 1, I've explained this already. Phase 1, you can see the egg entering the coronal radiata, the sperm entering the coronal radiata. Phase 2, the sperm contacts or enters the zona pellucida, which is this. Phase 3, the sperm enters, this is the cell membrane, plasma membrane of the egg cell. The sperm, phase 3, the sperm uses its acrosome layer or the head to enter the plasma membrane. Now, it now enters the cytoplasm of the female, of the egg, the female egg. Okay, so phase 2 has been explained here. When the head touches the zona pellucida, we call it the acrosome reaction takes place. That is, we all know the acrosome reaction I explained earlier on. So it will exclude other sperms from entering. That is what we call the acrosome reaction. And the acrosome reaction will, the head of the sperm will react with the zona pellucida so that it can get more way, it can get a free path or channel to go through the plasma membrane of the egg. So we call that one acrosome reaction. Then also phase 3 is when the cortical reaction, when the head touches the plasma membrane of the egg cell. So afterwards it enters inside and fertilization begins to take place. So other sperms are excluded from entering. That is what we call the cortical and zonal reaction. The zonal reaction is the acrosome reaction, the zona pellucida. And the cortical reaction is the prevention of the polyspermy. That's when it touches the head of the sperm touches the cell membrane of, or the plasma membrane of the egg cell. Other sperm should not come into contact with it. That is what we call cortical reaction. Okay, so this is what happens. We have a pronucleus, female and male pronucleus. They join together. The female one has 23 chromosomes. That's the haploid number. The male one has 23 chromosomes, haploid number. They join together to form a diploid chromosome. There are 46 chromosomes. So this is a general outlook on fertilization. So for one, sperm makes contact with the acrosome. This is the acrosome of the sperm, the green colored thing on the head of the sperm. It makes contact with the zona pellucida. Then number 2, the acrosome now releases hydrolytic enzymes which digest the walls of the zona pellucida. Then it's able now to enter the zona pellucida and react with, this is what, perivitelline space and also cortical granules are here. So now acrosome reacts with perivitelline space. It goes through the space. Then there are protein receptors here. The plasma membranes of the sperm and egg fuse. So the plasma membrane of the sperm and egg fuse. And when they fuse, the sperm nucleus enters the egg. Then cortical granules fuse with egg plasma membrane, which renders the replete layer impenetrable to sperm. So after cortical reaction is what happens when the sperm now enters, the head of the sperm or the nucleus enters the cytoplasm of the egg to prevent other sperms from entering, because it has entered. So that's the champion. All the other sperms, there is something we call game close. You have lost the competition. So go home. This is the champion. You will be able to fertilize the egg. So restoration of diploidy. So at this stage of the fertilization process, when the female and the male and female pronucleus or gametes fuse together, is when this is the stage where sex determination at the molecular level happens. Yes. So at this stage there is sex determination. Whether you will be a female or male, it is at this stage you will know. Then beginning of the segmentation. Okay, so now after this happens, the nucleuses fuse together to get the 46 chromosomes. Then the cell will now divide. The ovum will now start dividing into 2. Then the 2 will divide into 4, 4 will divide into 8, 8 will divide into 10, 8 will divide into 16, 16 will divide into 32. That is the blastocyst, and go and implant in the endometrium. So let's see another video. So like I said, I'm giving all credits to the owners of this video because they took enough time to prepare this video. So we thank them very much that they are helping us to educate our students. Let's continue with this video. And this video comes to explain the events in the first few days. So you can see the first week of pregnancy or embryonic development. The zygote becomes an embryo, then 2 cell stage, 4 cell stage, 8 cell stage, 16 cell stage, then 32 cell stage. The 32 cell stage will now go and implant in the endometrium. So let's see. And this is the egg, which is produced and goes to the ampulla region and joins the egg, the sperm here, to produce the zygote. So let's see what happens.
Welcome to my developmental biology playlist. In this video, we would look at the first week of embryonic development. So stay tuned till the end of this video and don't forget to subscribe. Let us look at the female reproductive system to understand the context of this process. In the female reproductive system, there are 2 ovaries on the 2 sides, and these ovaries Welcome to my developmental biology playlist. In this video, we would look at the first week of embryonic development. So stay tuned till the end of this video and don't forget to subscribe. Let us look at the female reproductive system to understand the context of this process. In the female reproductive system, there are 2 ovaries on the 2 sides, and these ovaries produce ovum or the female gamete, and this process is known as ovulation. In a separate video, I have described in details what is ovulation. So click on the video to understand more. Once ovulation happened, the ovum would try to migrate through the fallopian tube and it would reach the uterine cavity. Now imagine at the same time there was sperm present in the female reproductive tract. So it would try to fertilize this ovum. It turns out that there are 200 million sperms that is ejaculated in one round of ejaculation, but only 200 of them survives and make it to the oocyte. Out of these 200, only 1 would fertilize the ovum, and after the successful penetration, it would ensure other sperms cannot penetrate the ovum, and thus it prevents polysperm. There would be a separate video on polyspermy as well. All the links are in the I button. Now this penetration process is not easy because there is a thick coating of granulosa cells known as corona radiata. Sperm has to penetrate its way through the corona radiata to reach the nucleus. Then it has to penetrate zona pellucida. Once it has penetrated all these layers, it would eventually inject its genetic material in the cytoplasm of the ovum. Then the male and the female pronucleus would fuse together to form the zygote. Now, after the formation of zygote, if we wait for 30 hours, the cells would divide. A cleavage furrow would run through the entirety of the cell and dividing it into two individual cells. This is the process of cleavage, and it results in formation of two-cell stage. And this is happening at about 30 hours after the zygote formation. If we wait 60 hours, there would be 4-cell stage, 8-cell stage, and eventually after the day 3 to 4 there would be 16-cell stage, and at day 4 to day 5 there would be a 32-cell stage. One of the noticing factor in this particular process is all these events are happening while the embryo is moving towards the uterine cavity. Eventually in the uterine cavity, the embryo gets embedded into the uterine wall. Now while these divisions are happening, one important thing that we should understand, the cell number is increasing, but the cell size doesn't change that much. Even after the end of 8-cell stage, the cell size overall remains the same. Now at the 16-cell stage, which is known as morula because of its mulberry-like appearance, we can see the zona pellucida still covers the overall cells. It's like a bag that covers these blastomeres. After 16-cell stage, there would be 32-cell stage, and this structure is known as blastocyst. And this structure would be readily implanted in the uterine membrane or in the uterine wall. So blastocyst would shed its zona pellucida and get implanted into the uterine cavity. This would be the initiation of the second week of embryonic development, and this implantation takes place in day 7. If you want to learn more about the events of second week of development, click on the video in the I button right now. Click on the video in the I button.
So you can see this is the implantation site, that's the endometrium, the uterus. Either the anterior or posterior wall of the uterus, but most of the time the posterior wall of the uterus, that's in the endometrium. So this is the blastocyst. So the blastocyst will now, this is the outer cell layer, which is called cytotrophoblast, and the inner cell layer, which is called embryoblast. Then this aggregation of cells at the top of the fetal pole is what we call syncytial trophoblast, and the syncytial trophoblast will evade or erode into the endometrium so that this whole structure is called blastocyst. It can implant well inside the endometrium or the uterus. So it is entering it. You can see at first it was not entering, now it has entered. Then the cytotrophoblast has now succeeded. This is the cytotrophoblast. It has now succeeded in entering the whole of the endometrium. So there will be glandular proliferation, cellular proliferation, endometrial proliferation, and hyperplasia. There will be hyperplasia is the abnormal production of cells, abnormal multiplication or division of cells, so that it will now nourish this new this blastocyst. This is the baby, the blastocyst, the embryo, but at this stage it's a 32 cell stage, so we call it blastocyst. This is very small, a clot of blood in the woman's endometrium, in the woman's womb. A man is like a clot of blood, so this is a clot of blood. It has entered the womb, the uterus, and this whole process is what we call implantation. After implanting the uterus, you can see blood vessels. This is what we call lacunar system. Blood vessels from the mother. Blood vessels of the mother will be punctured, and when they are punctured, you get what we call the lacunar system. So, because the conceptus or the blastocyst has to be nourished. Let's continue the lecture. Segmentation or cleavage. Okay, so we will pause the video here for another class next week. Thank you.